Our next talk will be given by Dr. Shereen Ezzat, who is an endocrinologist and the head of endocrine oncology at the Princess Margaret Hospital, both a clinician who cares for patients, many of you in the room may know that, and also a scientist who runs a basic science research lab. Shereen is going to tell us about new advances in cancer genetics and predisposition to disease.
Thank you very much. So how do I go forward here?
Oh, okay. So you would do this or just the mouse?
Okay.
Mouse would be easier.
Thank you so much for inviting me. It's a great pleasure to be here. It's an even greater pleasure to have perhaps the most complicated part of a whole story to the very end and to leave it to me to try and put it together for you in 20 minutes. So let me just say at the outset that it's not going to be possible to do so. Instead, what I'd like to do is sort of put together for you some of the issues that have been discussed. And I want to put you sort of in the right framework. So the details may be somewhat fuzzy. And part of that is because they really are fuzzy. And let me see if I can get this to work here.
If you want to use this, yes,
this one here is not working.
This one. Take this.
Okay. So there are two aspects that I think I want to emphasize. This is not just a rarely diagnosed disease, but it also happens to be a fairly complicated disease. So it's not just a matter that there aren't too many people working on this like breast and prostate cancer. In fact, my lab is involved very much in breast cancer as well. And I can tell you this disease is probably more complicated at its root than even breast cancer, if I could even make that type of distinction. There are lots of diagnostic dilemmas, and there are diagnostic dilemmas because this cancer, as you know, although it is a solid tumor, we generally divide cancers into two major categories, what we call liquid, meaning it's in the bone marrow, or solid tumors, tumors that arise in the breast and prostate solid organs. I will try and make the thesis here, and this is somewhat speculative, but I think I'll try and produce some evidence here, that in fact this cancer, although it's solid, it behaves much more like hematologic malignancies, much more like chronic leukemia. And some of you are going to start raising your eyebrows, but hopefully that's what I want to do at 5:30 in the afternoon on Saturday afternoon. In fact, I'm going to put forward to you that many of the therapeutic conundrums that we actually face is largely based on the fact that we think of this as a solid cancer, but really it does not behave like solid cancer, and as such the treatments that we have used for solid cancers have not worked so well. And if anything, some of the drugs that are actually being developed for hematologic malignancies, ironically, are turning out to be potentially more effective. Is that really true? So in order to support this hypothesis, which is sort of a relatively broad theory here, I want to make sure that we're on the same wavelength, because you've already heard earlier that these are cancer-like. I think this is nonsense. We should probably eliminate that term entirely. This is a cancer. There are no other terms. It's a cancer. It just happens to arise from a hormone-producing cell, so it's an endocrine cancer. The histologic features I want to emphasize because if in fact we're going to make headways in terms of knowing who's more likely to develop the disease and who's more likely to respond to different types of therapies, we have to perform studies that are equally matched based on histologic features. Another major hurdle. Most of the studies in the literature actually are produced from very different centers that use very different criteria and apply basically apples and oranges together for comparisons, a major, major rate-limiting step. Similarly, if you're going to start to look at genetics, which basically requires that you bring patients and their tumors together, you have to make sure that you're talking about the same type of apples, or otherwise you're really coming up with the wrong diagnoses and you're trying to answer a question that's going to be built on the wrong evidence. And secondly, as you've heard from Dr. Shaw earlier, if you're trying to create a mouse model, how can you create something that authentically recapitulates something that you're not even sure what it is? So that becomes another paradox in this whole area. And obviously at the very end, what we ultimately have to do is be able to go back to our patients their tumors and the mouse models and make sure that they're all interconnected and that they're on the same wavelength. And that's been really our struggle, certainly in my clinic and in my lab over the last, I would say, the last 10 years in particular, where we've tried to look at this in a very serious way. You've already heard about this slide, that there seems to be sort of a curvilinear relationship, but I'm going to give you a different interpretation about this, again, as a clinician. The reason why there's such a long delay, it's not so much that a lot of doctors don't know about the disease and a lot of patients don't even know what to make of the symptoms. Because let's face it, if you're a patient, you get pain over the left side of your chest, you know it's likely to be potentially heart disease, and you go to the doctor and say, I think I've got something wrong with my heart. But if you got abdominal pain and some flushing and diarrhea, most of the time you're not even really sure what it is, and most of the time you're going to go to a doctor who's going to make the diagnosis of your bowel disease, which is exactly what happens here. But there's another explanation for this curvilinear relationship. It's actually got to do with the biology of the disease itself. The vast majority of patients with neuroendocrine tumors tend to have two phases to the disease. One is what I call the slow incubation period. That's generally in the order of decades. Dr. Olberg mentioned earlier that it's probably the time that it takes to reach more than 1 million cells before you're actually able to visibly see something on conventional imaging. That's, to some extent, that's probably true. I think actually that's an extrapolation based on mouse studies, if anything, because we know that when we inoculate 100,000 cells, we can't see anything. We have to get to 10 or 100 times that much to be able to see something, even on CT. But more importantly is that something happens with neuroendocrine tumors where there's a transition. And the transition takes it very much like chronic leukemia and transforms the disease from a chronic, stable, unrecognized entity to one that becomes much more accelerated. It undergoes transformation. And that is a well-characterized phenomenon, again, in chronic leukemia. And specifically, in chronic myelogenous leukemia, we know that patients have it for many years, and we know that there are some specific genetic abnormalities that occur to result in this switch or transformation from the slow to the accelerated phase. I would submit to you that one of the major challenges that we have is to be able to recognize when that takeoff point occurs. And I bet you that there is going to be a genetic hallmark that distinguishes those two very different phases of a disease. And to me, that I think is absolutely pivotal, because if we don't, we will not be able to tell the difference between those patients who probably need to be left alone with no treatment at all, to those in whom the disease is likely to become progressive, to those in whom progression has already started, the cat's out of a bag, and now we really have to initiate a different type of treatment paradigm. What's happened in most of the studies that we have in the literature have been based on a whole bunch of populations spanning the spectrum without clear separation of the two very distinct phases of a disease. The tumors, as you know, are basically from one orifice to the other, not just of the GI tracts. I want to emphasize something which has not been mentioned earlier here today, or perhaps I missed it, and that's in the respiratory tract. You can actually get all the way in the respiratory tract also endocrine cells, and when those endocrine cells are the focus or the nidus of a cancer, that is a neuroendocrine tumor of the respiratory tract, and we call that a pulmonary carcinoid. Probably the wrong term, but also, as has been mentioned, depending on the cell type, then you're going to get hormone production. Now, it's interesting, I happen to also have training in endocrinology as well as in oncology, and I would submit to you that the hormone aspect of this disease has created much more smoke and confusion than anything else. If anything, I would submit to you that most internists, surgeons, and oncologists probably become confused about this disease and try and shy away from it because they don't want to deal with hormones. Hormones is a bit of a black box. It's kind of like, wow, that's for hormone doctors. And it creates a lot of confusion because hormones tend to sort of elicit very different type of ideas away from cancer. And my view is that I think we need to separate this whole thing. that the hormonal aspect is really a relatively separate, independent component. This, as I said earlier, is a cancer and needs to be diagnosed and managed as a cancer with probably less attention to the hormonal aspect. And in many instances, what we have is diagnoses that are basically based on the hormonal aspect, and that is one cause of significant delay because we're waiting for patients to come with flushing, wheezing, and diarrhea to diagnose the carcinoid tumor that probably was growing for many years. And in fact, there is no carcinoid tumor. There's an endocrine cancer. It just happens to potentially produce serotonin in excess, but also it could produce gastrin, as you've heard, with recurrent peptic ulcerations, insulin, VIP, and so forth. In fact, if you look at the anatomic distribution of tumors, you find a reverse relationship, meaning that actually if you were to look at autopsy series, you find that neuroendocrine tumors are spread with very different proportions than what we see clinically, meaning that the two do not jive together. A lot of patients are probably misdiagnosed and probably not even diagnosed at all during life. So as a result, we have what we call well-differentiated neuroendocrine tumor as one terminology, the clinical syndrome of carcinoid, and somehow the two of them together are supposed to imply that there's going to be a prognostic implication. So sometimes we're lucky enough to find a tumor, we submit it to the pathologists, and sometimes all we have is a metastatic deposit. And most of the time, if we're really lucky and if we have a really good pathologist, they actually will perform the staining to tell the patient that this cancer in the liver is actually of neuroendocrine origin. And it's only when they start to twig into that that they will do the appropriate immunohistochemical markers. Sometimes the primary is not even obvious, and sometimes malignancy itself is not well appreciated, and the pathologist will refer to it as a well-differentiated neuroendocrine carcinoma. So these are the markers that typically people look for in order to make the diagnosis, and they also look for hormones to be able to determine whether it has the capacity to produce an excess of hormone. Again, a little bit of smoke, to be honest, not nearly as important as recognizing that there is a cancer in and by itself. So ultimately, let's get down to the nuts and bolts of it. Is it really going to be a bad cancer? So what people have tried to do in the past is look at the cells, try and determine if their DNA is actually undergoing rapid replication and separation. That's what the term, if you remember from high school, mitosis. Remember mitosis with bacteria? So those are the mitotic figures. That's the stain that often we use to label the separation of DNA material, and that's what the Ki-67 index is. It's an index of the proportion of cells in that specimen that's trying to tell you how much active DNA copying is going on for the purpose of separation to undergo mitosis. Okay? So that's what the Ki-67 is all about. It's useful when it's elevated, as you've already heard, but probably not terribly effective because the majority of samples do not give you a good representation of the entire tumor. So again, I'd like to move actually away from the Ki-67 story. I think it's passé, and to be honest, it's probably putting us in a bad category because it doesn't really give us good understanding. So the pathologists then, if they have enough specimen, they look for evidence of so-called invasion into the adjacent tissues, and typically around nerve bundles, which are intimately intertwined with the blood vessels, also in the muscle and the fat, and this degree of invasiveness, when present, also gives them a very good idea that this is likely to be a bad tumor. So in year 2000, the good minds who deal with this in the area of pathology sat together in Lyon and came up with a classification that tries to distinguish between the behavior of the good, the bad, and the ugly. And they called it benign, uncertain, meaning I have no idea what it is, and of course somewhere between a low-grade carcinoma, still with the idea that this is a different type of cancer. And they used the terms that we discussed earlier, which is the presence or absence of invasion to the surrounding structures, again the mitosis, remember the bacteria dividing, and the Ki-67 figure. So in other words, what they tried to do was come up with an integrated measure to classify the tumor into one of those two major categories. I would submit to you a relatively useful at the time, that was back in year 2000. In the 21st century, I think that this is very much behind. If we tried to apply this criteria for breast or prostate cancer, we'd be told stupid. So, what do we know about going back to the patients? What is it about genetics that's going to really help us? Well, we've defined two groups, and you've seen this slide earlier before, distinguishing the survival of patients who have less than 10% of their disease in the liver from those in whom the liver is occupied by more than 10% with cancer. And you can see in red that the survival really goes down when you've got lots of bad disease in your liver. So not really surprising. It means if you've got bad disease to begin with or at the time of diagnosis with evidence of significant liver involvement, you probably are not going to do very well. So again, in year 2000, remember when we were expecting all of the computers to stop working? That was also the end of the Genome Project. So the Genome Project was a roadmap, and this is what the Martian View of a cell looks like. Essentially, it's a series of electrical networks communicating information from the outside of a cell through the cell and into the nucleus. Remember, the nucleus is where all of the DNA material is. And those are the decisions that are going to be made to tell that cell whether it's going to remain alive, dormant, or whether in fact it's going to engage in replication and start to divide. So the information from the outside has to ultimately get down into the nucleus. So what we really have to do now is we got to be able to use this roadmap of all of the network hooking up the wiring system from the outside inside to be able to explain what the pathologists are saying so that we know what wiring system is being employed in that tumor. Okay? So, in order to do this, what we have to do is basically take all of the DNA from that tumor. And basically, one approach is to be able to perform what is called a microarray chip, which is basically a board that has all of the genes that have been characterized, allow the patient's tumor DNA to interact with this board and identify which genes are more likely to become overrepresented, more activated, from those that are underrepresented. Okay? So it's a relatively crude, but at the beginning, again, this was about 7 years ago, everybody thought the chip was going to be ideal. It was going to be the way to spend $10,000 on a sample and get very good information. Luckily, by now, we can now spend $1,000 and get that same information. But what it implies is that the overrepresentation of that gene is necessary and sufficient for the development of this patient's cancer, and that indeed this gene is instrumental in maintaining that cancer. Because remember, the purpose of this exercise is not just to know how it started, but how it's surviving and how we're going to specifically target certain pathways. Very critical. So let's look at the specific genes now. And let me introduce you to your parents. That's your mother and your father. Can you see your mother here? She's in blue. And your father, giving you the full complement of genes. Remember, you have 22 of them, and of course the 23rd is for X chromosome information, genetic information that's related mostly to just gender. But the 22 autosomes represent information that includes all of the genes that we've talked about. Now the problem is that there's lots of other information that occurs between these genes. And what we had hoped is that most cancers would fall into the category where a single defect, as shown for example here, would represent a hot spot, so that there is an inherited defect passed on from your mother to you, and that you would demonstrate the presence of a disease on the basis of this single defect. However, this turned out not to be the case. Very few conditions fall into that category where there's a single gene that causes the disease. So the other possibility is that maybe the father also contributed to the disease. In other words, that there's a dual defect, that you inherit a defect from your mother and a defect from your father. And the nature of the defect, as you can see here from the X's and the O's, does not need to be exactly the same. In other words, each parent will give you a different type of defect towards contributing to an abnormality for that particular chromosomal region and DNA material that goes along with it. I'm going to be coming back because sometimes some of these defects do not represent abnormalities of qualitative nature, but they can be quantitative. For example, what you're seeing here in red, where part of a chromosome is actually gone. It's deleted. And that, you can imagine, losing that information can be problematic. And in the case of neuroendocrine tumors, we've known for a long time that there is loss of material on chromosome 11, the long arm of chromosome 11. And specifically, after year 2000, we were able to know that there was a gene in this region called menin. Has nothing to do with men. It's multiple endocrine neoplasia. And individuals who have a genetic predisposition, they carry a defect in this gene, which is transmitted, and predisposes you to develop neuroendocrine tumors, but also as part and parcel, you also develop tumors in other parts of the body, including the pituitary and the parathyroid. I should mention, however, that there are probably multiple other genes on the long arm of chromosome 11 that have not been characterized, also associated with multiple endocrine neoplasias as well as pancreatic neuroendocrine tumors. There is also a condition called von Hippel-Lindau, VHL. I don't know if there's anybody in this room who has it or is aware of it, but VHL is also a defect characterized by a heritable transmission of a gene that predisposes to typically pancreatic neuroendocrine tumors, but also they get defects in the brain, in the retinas, and possibly in the adrenals as well. And this needs to be identified because it alerts the individual not just to the presence of one, but possibly multiple tumors in the same individual. And more importantly, because it is what is referred to as autosomal dominant, it is transmitted from one generation to the next. So that's an example of what we refer to as a heritable defect, transmitted from one offspring to the next. And these are examples of such defects with syndromes well characterized, I already referred to as multiple endocrine neoplasia type 1, or MEN1. The next are defects that are not present in your germline, meaning not in every part of your body, but is present only in the tumor. Okay? So these are what we refer to as somatic abnormalities, characteristic only to the tumor cells. And they're important to define because in some situations, the patients present with disease that is referred to as sporadic. Sporadic means you're the single lucky member of that family who happens to have a disease. And initially what people did was they went and they screened for abnormalities in the tumors, in the genes that were characterized in other types of cancers. That was a very logical thing to say. For example, if you have a neuroendocrine tumor of the gut, what about garden variety gut cancer, colon cancer? And that's what Dr. Shaw was referring to earlier, P53, but unfortunately it turned out not to be the case. And instead, there seems to be abnormalities at multiple other genes in neuroendocrine tumors, suggesting that we're probably dealing with a heterogeneous group of individuals with multiple types of defects, indicating that in all likelihood there isn't going to be a single gene responsible for all of the diseases that we've been talking about here today, but there are probably going to be multiple types of defects with different importance for each patient's individual tumor. And what we already know now, that for example, if you look and you compare the primary tumor from a metastatic tumor, we're starting to see some patterns. So for example, if you have primary lung cancer of a neuroendocrine nature, it's much more likely to show abnormalities again on chromosome 11 as compared to gut neuroendocrine tumors that are much more likely to show abnormalities on chromosome 18 instead, suggesting that there is a chromosomal separation of the different sets of genes that are affected depending on where the cancer arises. So to define the specific genetic defects with greater certainty, what my lab has tried to do, and I'm going to show you this, and I want to emphasize that while we are very enthusiastic in my lab, and I'm going to show you some what I think is impressive data, it's a very, very small part of the big picture, okay? So let's just be very focused on this. These are the cell surface receptors that most people have been focusing on in the last 10 years in terms of cancer progression in particular. Cancer progression genes are genes that basically turn very small incidental cancers to big deal type of cancers. And for example, the IGF-1 receptor is a progression cancer in prostate disease. It is not the case in neuroendocrine cancer. My lab is focused on a different type of cell surface receptor called the FGF receptor, and the reason for that is we knew that if you knock out this gene in the mouse, the single most prominent feature that you see is abnormalities in the hormone-producing cells of the body. So it gave us an idea that from a genetic point of view, this type of receptor is important for hormone-producing cells of the body in terms of their ability to differentiate and to grow. Okay, somehow it doesn't want me to. Can I get you to do the next one for me, please? So what we did is we started to look at this family of receptors. The FGF receptors unfortunately turn out in humans not to have one, but there are 4 major families on different chromosomes. And the one that we started to focus on some more was the one on the very far right, FGFR4 specifically, because it seemed to be much more overrepresented in patients who have neuroendocrine tumors, particularly those arising from the pancreas. If I could have the next one, please. And this is a slide actually that is borrowed from Stefano Serra, who's sitting in the back, and several of the slides that you're going to see next. Stefano is now a young pathologist who's finished his training with us at UHN and is going to be a staff pathologist looking at many of these tumors. And what he was doing was really looking in his project at an abnormality in the FGFR4 gene. And what you see here is the term polymorphism, and I want to introduce you to this term. Polymorphism means different shape, meaning that we all don't have exactly the same DNA sequence for this gene. Half of us roughly have a neutral form of this receptor, and the other half of us happen to have a charged form of a receptor, so that the sequence changes, causing a single substitution from a glycine, which is a neutral amino acid, to an arginine, which is a charged amino acid. Single letter change in the DNA material, as you'll find out, causes a huge difference. Now, why do we call this a polymorphism? Because it is very frequent in the overall population. It is not just present in the tumor cells, but it's also present in the rest of the cells of our body. Okay, maybe, yeah. So what he did was he sequenced the DNA material from patients who had pancreatic neuroendocrine tumors and divided them into those that are we call glycine on the left, which is the wild type or the normal, and I will call glycine good. And arginine, I don't know what I'm going to call arginine, but it's bad, as you'll find out. Next. It should work now? Okay. What we've learned when we started putting this DNA material together is that this single base substitution in the receptor causes very different wiring of the information from the outside of a cell into the nucleus. So that while the glycine, the good, tends to favor this pathway, this is a pathway that is referred to as the MAP kinase pathway in neuroendocrine cells that actually promotes the cells to remain behaved and produce hormone cells as they were taught to. However, when they become more acidic up here, the conformation, the shape of a receptor changes enough that it starts to now communicate information to the nucleus through a different pathway, one that involves the so-called PI3 kinase AKT pathway that promotes cells, A, to survive, and also allows for activation of a pathway that makes cells start to float around, just like little amoeba, having the potential to metastasize, away from the good and closer towards the bad. Why is this important? Because to us, this is absolutely crucial in defining which drugs you're going to use, not just in an experimental system, but in humans. Many of the drugs that you've heard about today are very selective to specific pathways. And unless your tumor happens to rely on this pathway, this drug is only going to cause side effects without good effectiveness. More importantly, as I mentioned earlier, remember there were two phases to the disease, the slow and the progressive. If you're not dealing with the disease at the right time in the right place with the right drug to interrupt the right pathway, you're probably causing more side effects than effectiveness. So that's why this was so crucial for us. So I have to emphasize, by the way, there is precedence for this type of abnormality in breast cancer, not for this gene, but in something that you may have heard about before called HER2/neu. HER2/neu also has an abnormality also in the same region, the corresponding region, that changes the conformation of a receptor and starts to create a very different pathway that promotes bad breast cancer progression. So this is, again, a biochemical paradigm that nature has already used in a different type of disease. And again, just showing what Stefano did is when he started to look at the genotypes and used all of the classical parameters that the WHO told him should predict bad disease, what he did find is that in fact patients who had disease that was more likely to spread to the liver had more of the arginine instead of the glycine form of the receptor. And in order to determine whether this really was true, and this was not just a figment of our imagination in terms of the signaling work that we do in our lab to satisfy reviewers of a scientific nature, what he did was actually develop a different type of orthotopic mouse model similar to Dr. Shah's, except this one, we insist not on introducing the cancer into the spleen because we don't want it to grow into the spleen. We want it to actually to allow it to be able to give it the propensity to create its new home. And using this approach, again, what he was able to determine is on autopsies that if you involve more of the bad arginine instead of the glycine, so what he's able to do is he's able to engineer the cells and change them from their wild-type state to the arginine. And he has a comparison of the two. And what he's able to do is demonstrate that if the mouse is carrying cells that have more of a charge, the bad type of receptor, that the cancer actually starts to grow and grow into the pancreas. And as you can see, down into the abdomen of the mouse. If I could have the next one. But more importantly, you can see the tumor visibly with multiple metastases grossly. And more importantly, he can identify the metastases at the time of autopsy and identify their size as well as their cellular origin. But we wanted to go one step further, and just like Dr. Shah, we wanted to be able to keep our mice alive. We didn't want to sacrifice our mice. More importantly, we wanted to be able to determine what the natural history of a disease was going to be. Now, what you're seeing here is Stefano's mouse undergoing a CT scan. We don't use a luciferase. We actually use the real thing. It's a micro-CT. The mouse has to lie down, be still, not move, show up for the appointment on time, and watch the tumor now growing. And you can see what happens. So that was a normal mouse. Now you see this mouse is developing metastasis after about 2.5 weeks with tumor development into the liver. This is actual real live imaging of a live mouse. More importantly, we're now performing 3D reconstruction. So we can get not just two-dimensional, but three-dimensional volumetric assessment of the tumor burden. So we can quantify not a surrogate marker of a disease, but the actual disease itself. And in this case, we can come up with a quantitative measure of the tumor weight in the mouse. We can then subject it to different types of treatments. And this drug actually, and that was used in this particular experiment, is wabane. Wabane is a member of the digitalis family. This was in other types of experiments done by another member of our collaborators, where basically we discovered that digoxin, a drug that's used for heart disease, turns out to have anti-cancer properties, and specifically in this disease. And it does so by interfering with the ability of a receptor to get to the cell surface. So if you recall the size of a tumor before, and this is now using digoxin itself in a mouse exactly treated with a bad carrying cancer and allowing for the mouse to be able to live and take the drug and measure that the tumor is actually getting smaller and remaining that way, as well as the metastases, which we can also perform PET-CT on so that we can look at the metabolic activity in addition to CT imaging. However, if you thought luciferase was expensive, try and get an appointment for a mouse where, again, the hospital charges $1,000 per CT scan with an appointment ahead of time. Many of you, I'm sure, are aware of that.
Hospital doesn't allow.
Does not
I can't put my mouse with the patient's table. That's it.
I'm glad that at least they got their priorities straight. But here in Ontario, $1's a dollar. Finally, I just want to emphasize that this is just one example of what really can be done and is actually being done in other types of cancers. But more importantly, I want to emphasize something else, and that is something that goes beyond genetics. Something that I think is getting closer to what I think this disease is all about, something that we call epigenetics. Classically, when people talk about cancer, they talk about genetic abnormalities, and we talked about some of these mutations or so-called polymorphisms depending on the frequency in the population. We talked about the loss of genetic material. We talked about deletion of genetic material. But something else can also happen, and that is called epigenetic. Epi meaning above. Above. What controls the genes? Because remember, every boss has a boss. And it turns out that there are, in fact, modes by which the control information that directly allows for this gene to either become active or silenced is also subject to control. Meaning you can have the bad gene with a mutation in it. But if that gene is silenced, this gene is null and void. It's like a CEO who's gone to retirement. Who cares? But bring them back from retirement, resurfacing them is possible by means of DNA methylation inhibition. And in fact, one of the drugs you've already heard about, temozolomide, acts through this mechanism. Is by inhibiting DNA methylation. The problem is it was designed specifically, actually initially for CML, thank you, but more importantly that there are other modes of DNA control of access to information that is referred to as histone modifications. Histone modifications are even more complex, and in the end, they're modes by which the DNA is actually compacted and associated with different proteins that regulate access to the DNA information. It's a much more complex story. I believe the future is there because I think that just because you happen to have a genetic defect is not a be-all, end-all. There are also modes by which genetic information can be either silenced or activated. And I think it's going to be clear that drugs specifically targeting these pathways in a very selective manner are going to be absolutely crucial. Because these type of modifications, as shown in these slides just very quickly, to show you they can actually be regulated. Ultimately, this is a theory. This is really what we're trying to come and bring together, is that age, the environment, the DNA genetic information, as well as the parental information that imposes on genetic information are ultimately captured in a global computer program that you carry with you, that responds and adapts itself to this information in a manner that integrates also your genetic background to ultimately determine what disease you're going to get, when you're going to get it, and how it's going to behave. It's a long toll, but I think that this is a much more integrated way rather than being purist about just genetics or epigenetics or both. I want to end by listing the collaborators that we have in Europe as well as those who work with me in the lab, including Stefano. I've shown much of his data here today. And more importantly, I just want to emphasize, and this is really a disclosure, I happen to live also with a pathologist. And this is a very tough toll because she constantly keeps telling me that I'm not on the mark and I have to get closer and closer to what happens in real life. Thank you so much for your attention.
Well, that concludes our formal talks for today. I know many of you probably have to get home. I guess I'll ask Maureen and Jim to tell us whether we have time for any questions at this point, or should we just take all the questions and do them either tomorrow or online?
First of all, before we do the questions, to answer your question, this is for you, Sylvia.
Oh my goodness. Thank you very much.
Thank you so much for doing the 8.5 hours of CME credit through the University of Toronto.
We'll do those after the questions.
First the clapping, right?
We'll do those after the questions.
Pardon? We'll do after the questions?
That's right.
Questions first, a couple of housekeeping things after that.
This one is for Dr. Oberg. Where are the gaps in imaging knowledge? What research should be done? And how could CNETS Canada best support that?
Can I use this? You hear me? Good. I think there are a lot of gaps still in molecular imaging, and we had to solve that out. And of course, we need more support from different organizations. And we had to learn more about the expression of different receptors on the tumors. We had to learn more about how actually each type of tracer is accumulated. And of course, we had also to look into the future, which markers, which tracers should we be using. So we have just started on the trail, and I think there is a lot of more to be done. And in 10 years' time, I think a lot of people will laugh at us and say, why did you use so bad markers in the beginning, so bad traces? So I think we can be more sophisticated. And I think the development is now started. And I think money and knowledge and chemistry and all that stuff are lacking in many centers.
Okay, thank you. Supplementary to that, a number of questions have come in about imaging that I will attempt to answer for you. First one is, where are PET scans available in Ontario? Well, the good news is that there are PET scanners scattered around Ontario. There are actually a couple of them in Ottawa, in Toronto, Hamilton, London, Thunder Bay, and I think there will be more. The bad news is that we've been funded, well, the good news is we are funded to do a very narrow range of PET applications since the first of this month. And we're allowed to do PET imaging on patients with lung cancers, colorectal cancers, lymphomas, thyroid and germ cell cancers, and I think esophageal. And that's it. Neuroendocrine is not on the list. You can try. There is an appeals process, and your doctor is the person who would make an application to say, can we make an exception for this person? I cannot tell you how many hours. I estimated once that I'd spent 1,000 hours in the last 10 years trying to advance the cause of PET scanning in this province, and we're behind many developing countries in respect of the evolution of this technology. The ministry says until we have class 1 evidence for every indication, we're not going to pay for it. So there is no room apparently for physician judgment in this matter. So if your physicians feel that you would benefit from a PET scan based on the evidence in the international literature, all I ask you to do is pound the doors, because a number of us have tried very hard and we're exhausted from the attempt. There's another question here about the availability of octreotide scans, and they're recognizing that they're not available in every hospital. And this is, again, an issue of funding. We have been doing octreotide scans in London through a bureaucratic wrinkle that I don't need to explain. But many other hospitals are not able to do them because they haven't got any money in their budget for octreotide. And it is, after all, in a general hospital, it would be a rare exam. It would cost over $1,000 for one dose of octreotide for a diagnostic study. The worst piece of news is that if you live outside our LIN, you shouldn't come to us because we've been told we can't deal with neuroendocrine tumors from outside our geographic area. So again, you need to pound on doors. And emphasize, choose your targets, emphasize to the people who are politically influential that this is unsatisfactory and is not meeting any kind of international standard. I'll stand behind that if you want to quote me. Okay, we have a quick question for Dr. Ezzat, if he's still in the room. The questioner is asking if genetic testing for carcinoid is available or appropriate.
There are two major genes that I think most patients should be screened for, but only under specific circumstances. I don't think we're at a point now where we can apply it to everybody, but specifically if you happen to have more than one tumor in your body, if you happen to have more than one tumor in the endocrine system, and if you happen to have more than one family member affected by an endocrine tumor, then such individuals should be tested for a genetic abnormality. And the two that are most important in the blood would be those for MEN1, which is done here in Ontario, and the other would be VHL, which actually is sent to another lab in Calgary. But again, that depends on the type of tumors that you have, where they're located, and whether there's more than one family member affected. So the medullary thyroid would really be in those patients who have medullary thyroid cancer, but not necessarily the pancreatic or the gut that we've been talking about.
Thank you. There's a last question that perhaps any doctor in the room might want to contribute to. I'll read it and say something about it, and then we'll be open for comments. The question is, what are doctors doing in terms of their continuing medical education to stay up to date with the algorithms for diagnosing, treating, and following carcinoid tumors? I think you've just seen a minute ago one piece of evidence that the doctors who attended today's session are getting some continuing medical education credits for that. We're all obliged to satisfy the Royal College or the College of Family Physicians to acquire CME continuous medical education credits for a certain minimum number of hours every year in order to maintain our certification, our license to practice. That doesn't say that anybody has to choose neuroendocrine tumors, of course. They can choose other topics. Does anyone else in the room want to comment on the matter of physician education, maintenance of competence? Dr. Oberg.
I would like to welcome all our colleagues to the ENETS meeting in Europe. Every year we have the European Neuroendocrine Tumor Society meeting, and next year it's in Berlin in March, and I welcome all of you. Some of you are also welcome to my course, which is running every year. This year is for the 16th time.
Thank you very much. And just one last question, which has a short answer. It's also directed to me. Why is the McMaster reactor not covered to manufacture isotopes? Well, the McMaster reactor may be part of the intermediate term solution. They're talking about tarting it up and making it available to produce possibly some of Canada's needs for molybdenum technetium, but it won't be producing, as far as I can tell, other isotopes such as yttrium or lutetium or gallium. The McMaster reactor is also 50 years old, so it's not it cannot be part of a long-term solution. And so we're just still in that straitjacket. All those issues are being looked at. And with that, I think we have dealt with all the... If your questions weren't read out today, they are not less valuable. They will appear, they will be assigned to someone to answer and appear, I guess, on your website. Thank you.